Hi, my name is Bridget Maloney, and today I will be presenting on Phylum Orthonectida. When this phylum was first discovered in 1877, it was believed to be grouped with the protists. However, it's now understood to be a transition group between Metazoa and Parazoa, which is why it's placed here between Platyhelminthes and the annelids and mollusks. What all of these groups have in common are their spiralian characteristics. They're all protostomes and bilaterally symmetrical. So a general body plan and an overview of our orthonectid. Like I said, they were discovered in 1877, and 21 known species have been discovered since then. They're within 4 different genera: Rhophilura, Intishia, Ciliocincta, and Stocarthrum. So the general morphology of the orthonectid, we can see the muscular system running through longitudinally this way, and then we have the gonads and oocytes that are in this cluster at the bottom. The winding digestive tract, compared to an adult protostome that is bilaterally symmetrical, you can see the gonads are much larger and just a much different setup, much more compact in the orthonectid. There are no body cavities, organs, a gut, or a nervous system within the orthonectids, and they're generally less than 1 millimeter long. All of them have a one-layered cuticle, except for one species, Intishia linnae, that has a 2-layered cuticle. So this is a picture. You can see the cilia covering the outer cuticle, which is their primary means of locomotion. They are free-swimming and ciliated. They move in a purposeful direction, which is aided by these circular muscles. They can either swim in a straight line or twist and bend depending on the environment or host that they're in. The outer layer of cells are contractile rings that act as muscle. This is very similar to the annelids and polychaetes that we've seen before that use this type of muscle to move. And we can see the bending and twisting movements down here versus the straight line swimming. So the genome of Intishia linnae is the main genome studied in this group. It is the 3rd smallest genome in the animal kingdom and has the lowest number of transcription factors necessary to maintain the bilateral symmetry that we see in this group. A quarter of the genome is constituted of unclassified, segmented, repetitive cells, very similar to what we have seen in other groups, including the annelids and the polychaetes. Hox genes are not organized in a cluster like they usually are. Many are missing or replaced by neural genes. So reproduction within the orthonectids, both male and female, they're gonochoristic generally, will have genital pores, which is where internal fertilization happens. The gametes are surrounded by a layer of smooth muscle and cilia. Stocarthrum is our only genre that is hermaphroditic, and Rhophilura ophiocome is our organism that has 2 different types of female body plans. So Rhophilura ophiocome, this is the plasmodial stage. You can see it's multinucleated, and instead of just one cell with one nucleus, it's scattered throughout the plasmodium. You can see the developing embryos and the agametes all within this stage. And then looking closer, you can see the plasmodial cells developing into sexual adults, the gametes developing within. And then you can see the much larger female on the right and the much smaller male. These are the mating adults. And that is the infective larval stage that comes after. So the plasmodium is the development of orthonectids. It's a cytoplasm that is multinucleated rather than a single nucleus in a single cell. And a general overview of the life cycle. There are both asexual and sexual components, but the majority of production of sperm and eggs, the zygote, fertilization, and the ciliated larva all happen sexually, and fragmentation occurs once the orthonectid has already entered its host, and that's where the fragmentation and asexual reproduction components play. Like we said, orthonectids are marine parasites. They will parasitize brittle stars, mollusks, flatworms, ribbonworms, ascidians, and the basic mechanism by which they do this is that the larva will disintegrate, the ciliated layer will be completely shed within the host, and the inner orthonectid cells will then scatter into the host tissues and become infective cells. That'll hypertrophy the muscle and completely disintegrate it. So these are just a few different relationships between orthonectids and what they parasitize. And over here we have 2 different examples, but what we're going to focus on, as you can see, with the flatworm infected with plasmodium, these are the small plasmodium, much smaller than the actual flatworm. And the relationship that we're focusing on are the Rhophilura ophiocome and the Amphibolus squamata. So that is a brittle star that the orthonectid is infecting. The male orthonectids will die as the females continue to incubate their eggs until they become free-swimming larvae. The larva will then enter muscle cells and the bursal pocket of the brittle star, which is where they incubate their eggs, or it'll enter their sac-like gut, and it usually will exit through the bursal pouch or through the mouth. The muscle cell of the organism becomes hypertrophied, and the parasitic cells will become massed clusters in the cytoplasm. So this is the brittle star species that we're talking about, and this is our Rophilora. You can see the female is, again, much, much larger than the male. And here, another comparison. You can see we have two different female body types, or body plans, over on this side, and then again, the much different looking males. This is just a close-up of a hypertrophy contractile cell in that brittle star Amphibulus squamata. You can see the outer covering of the muscle cell, which is through which the parasite will enter, and the developing embryos will hypertrophy the cell. And then this is the epithelium of the gut, which again is where they can enter. And you can see the multinucleated cells, developing right inside. These are the clusters of germinal cells all around the edges, and it's the early development of a female Rophilora ophiocome. Final thoughts. Looking at this genre, it became very evident that it is highly understudied, and looking through many vertebrate parasitology books, it's clear that parasites that affect invertebrates are already understudied. In this particular phylum, there are just a couple of case studies, many done 40 years ago, that are still, constantly being revised and changed, and much more needs to be learned about them. But it is very clear that their reduced body plan makes it much easier for them to survive as such a successful parasite, and they're still classified very close to these really complex groups because the features that they originally had that made them so complex have been lost over time due to their ability to parasitize so effectively.